Hi everyone, welcome back. So we're going to be continuing our cell cycle notes. So hopefully you've been keeping up and you've learned about how DNA is packaged so that way it can fit into the nucleus. Some of our specialized proteins called histones that allow our DNA to wrap up and eventually form something called a chromosome. We've talked about diploid and haploid cells. And then we also talked about the cell cycle, which is a process that cells are going to go through whenever they need to reproduce, replace, or repair any of their cells. So if any of that sounds unfamiliar to you, I recommend that you go back to last week's notes, you go ahead and watch the video and fill out your notes from there. So let's go ahead and get started. We're going to go ahead and move on to our M phase of the cell cycle, particularly the phases that refer to mitosis. So during our cell cycle, our cell is growing, our DNA has replicated, we've doubled our organelles, and so now we're ready to divide. So for mitosis, we actually are going to go through and divide the nucleus first. So I went ahead and added an extra text box up here. So if y'all can go ahead, you can either copy and paste one of these text boxes or just form one yourself. And so right here, I wanted to go ahead and write the definition of mitosis. Okay, so the definition of mitosis is the process to divide the cell's nucleus. Okay, so we have double the DNA. We need to make sure, because we're trying to double our cells, so we need to make sure each cell has equal amounts of DNA. And so there's 4 steps to this, and we refer to those steps as PMAT. So first we have prophase, then we have metaphase, third is anaphase, and fourth is telophase. So we're going to go through each of these steps. That way you can go ahead and fill these out and figure out what your cells are doing during this time. So first for prophase. Prophase is actually the phase that the most things are going to occur. So if we look at that picture, one, we can see that we have chromosomes inside the nucleus. And so our first thing is that chromosomes form inside the nucleus. I think I'm going to run out of room. Let me go ahead and change this. So I'm going to make this into bullet points right here. So I encourage you to do the same. I'm also going to make this a little smaller. Okay. All right. So let's go back to full screen. Okay, so chromosomes are going to form inside of the nucleus. And if you look at the nucleus, you can actually see that the outside of it has these dotted lines. And so my second thing, my screen froze, there we go, is that my nuclear membrane is starting to dissolve. So additionally, if I look at these organelles, so I can see I have some organelles here like my endoplasmic reticulum and some mitochondria, but I have these little orange sticks right here. Okay, those orange sticks are called centrioles. They're always in pairs, so you can see there's 2 little sticks together. And so these centrioles are going to move to opposite ends, and that's because they help the cell to perform mitosis. And then lastly, you could see those little centrioles, those little orange things, have little blue string looking things coming out of them. Those are spindle fibers. So our spindle fibers are going to be used to help separate the DNA. Okay, so those 4 things happen during prophase. Prophase, like I said, is the most complicated, and that's when you have quite a bit going on. So after prophase, let's go ahead and look at our metaphase cell here. So you can see right here I have my chromosomes, which are my X's, and they're all lined up in the middle. I don't have a nucleus anymore. That membrane dissolved. So all of my DNA is now located in these chromosomes. And you could see the centrioles are on either side, and my spindle fibers got really, really long. And so what's going to happen here is that these chromosomes, because they're replicated DNA, my spindle fibers will attach to those chromosomes. That way it can separate the replicated DNA, so that way you have equal amounts of DNA on each side. So during metaphase, your chromosomes line up in the middle and attach to the spindle fibers. Okay. And we can see that right there. We say metaphase, M stands for middle. That's one of the easy ways to remember that. So now that they're in the middle, we need to pull them apart. We need to separate these chromosomes. That's where anaphase comes into play. So if I look at my anaphase, I can see my cell is starting to get a little bit longer, a little bit skinnier, and those spindle fibers are getting shorter. But in the process of those spindle fibers getting shorter, I could see my chromosomes separated in half. So for anaphase, my chromosomes are pulled apart to opposite ends of the cell. So now I have equal DNA on each side of the cell. So my last thing is telophase. I need to form two cells, and I need to also reform my nucleus, which you could see right here. So for telophase, I'm going to reform the nucleus, and then cytokinesis is going to start, where I either have that cleavage furrow, so my cell membrane is going to pinch in, or I'm going to form a cell plate in the middle if it's a plant cell. I can also look at my chromosomes here, and I can see it looks kind of, spotted. It looks like there's a bunch of little dots there. And that's because these chromosomes actually need to go back into chromatin. So remember how I talked about in the microscope, you don't see little X's in the nucleus. Usually you just see like a big blob of DNA. And that's because it's in chromatin form. So our chromosomes are going to unwind a little bit and they're going to form chromatin. So during telophase, and I'm actually, going to move this up because I know it's hard to see with my little camera there, and I can't move my camera. There we go. All right, so in telophase, my nucleus reforms, chromosomes turn back to chromatin. Okay, and then it'll lead into cytokinesis. All right, so hopefully you can go ahead and get that. Okay, so you can, you don't have to move your text box. Just go ahead and type that in right there. So what does this look like? So, how can we imagine this? How do we, how can we see that this happens? So what we're going to go ahead and do is I'm going to go ahead and show you this quick little diagram. That way you can kind of see what steps the cells are going to go through. So first we have two cells. I think because we're at the end, I had already played this once. So let's see what happens. Okay, it happened really fast. Okay, I'm going to go ahead and play it one more time. Okay, so let's go step by step. So first, we're at interphase. So you can see you have a nucleus here. Okay, I don't have chromosomes inside my nucleus, and I have my centrioles right at the top. They're not on the opposite ends. And right here, I have a little microscope slide, so that way I can see this is actually what a cell looks like under a microscope. And there's my nucleus. So after interphase, once we've replicated our DNA and our cell has grown, we're going to go into prophase. So in prophase, you can see the centrioles are starting to move. And we also start to have some spindle fibers. I no longer have my nucleus there. It's starting to dissolve, and I'm starting to get chromosomes instead of chromatin. I know they look really funny right now. Okay? And I can see in the microscope my cells kind of doing the same thing. I don't have a nucleus anymore. It looks very spotty, and my cells starting to get a little bit long and skinny. So prophase can be extended into prometaphase. Okay, so that just means, prophase is a little bit long and there's a couple of steps that can happen. So I can see now I have my spindle fibers, I have my chromosomes, and they're starting to move to the middle. So now we're in metaphase. They're in the middle. My spindle fibers are attached, and look, even on the microscope, that's what they look like. It's really hard to see, but there's a little line right there for my chromosomes. Next is anaphase. They pull apart. Okay, so now my spindle fibers are pulling the chromosomes just like it is in my microscope picture. Again, really hard to see, but they are being pulled apart. Telophase, you can see it's starting to pinch in a little bit. Same thing on the microscope. And then I'm going to start to reform my nucleus. Cytokinesis is when I have that cleavage furrow, and you could see my nucleus, or nuclei rather, because I have two cells, are starting to reform, even in my microscope picture. And now the cells are little baby cells, they're back in interphase, and they're going to do this all over again. Okay, so I'm going to play this one more time. All right, so hopefully that helps you understand mitosis a little bit. So let's go ahead. Okay, so in our notes, we already talked about mitosis. So now let's go ahead and go over some vocab. So we have some matching here, okay, and some of these you probably already know. So if I look right here, okay, so these are our centrioles. I know they're not orange, but they look like, I don't know, they look like little Twizzlers or little churros. So these are our centrioles. I'm going to go ahead and click this and put it over here. Okay, those are my centrioles. Remember these little, string things are my spindle fibers. Right here, okay, so I have a chromosome right here. So if you're confused about the difference between chromosome and chromatid, I'll go ahead and explain that really quick. So chromosome has your full genetic information right here. So like, for example, normally in our cells, we have our full set of genetic information in a chromosome. All right, right here in the middle, okay, that's our centromere. Okay, so our chromosome has two branches. It has two arms. It has the one at the top and the one on the bottom. When we're talking about chromatids, okay, so chromatids are when we've replicated our DNA and now we have two halves of this chromosome. Okay, so you could see you have a chromatid. You have one on the left and you have one on the right. Sometimes we call those sister chromatids. And so this right here is the duplicated chromosome. Okay. All right, and so what phase of mitosis is this one right here? Okay, very good, it's metaphase. Okay. All right, so I have one more thing for y'all. So in case you're having trouble remembering the steps for mitosis, we actually have kind of like a little cheer that will help you on. Okay, so first, okay, you're gonna put your hands together. Okay, so I just went ahead and stacked my hands like this. Okay, so right here, okay, so this is when my cell is in interphase. So I have a nucleus that's in a circle. That's because there's chromatin on the inside. This is interphase. Okay, so prophase, I'm gonna go ahead and cross my fingers, okay, but I still have kind of a circle. It's kind of loose. Okay, so this is when my nuclear membranes start to dissolve, but I start to get chromosomes, which are my little X's. Okay, so prophase, metaphase, my chromosomes are now in the middle. Anaphase, they're starting to pull apart. And telophase, they're starting to form two cells. Okay, so in case you didn't get that, interphase, I have a nucleus. Prophase, I'm starting to get chromosomes, okay, but my nucleus is still kind of there. Metaphase, they're in the middle. Anaphase, they're pulling apart. And telophase, I have two. So hopefully this helps. Let me know if you have any questions, and I hope you'll have a great day.